Welcome to Study Time, a televised home learning program produced by Rwanda Basic Education Board.
Hello viewers, welcome to the TV learning series. Here we are with the Senior Sixes, Gene Technology. That's the unit. So in today's session, we are going to look at profiling. So what is to profile? So we need to start thinking. Are there times when we have accidentally been taken to prison or judged wrongly and the judgment is passed and we spend 10 years in prison, then 10 years after, we are actually found innocent? So to the judges and those that do give justice to those that deserve it, how much of the proof do we have that someone has committed a crime? Or how much of the proof do we have that a child belongs to you, not to your brother or your cousin? They normally tell us that the only person who knows the father of the child is the mother. Do we all agree? And there are other ways we can do that. So, to the slide, then we check what we have. So, if you look at the background picture in the slide, the things we are supposed to look at. One, the objectives are that we should be in position to describe the process of DNA profiling. Can we write out, can we extract, can we read the information in the genes on paper so that we're able to know who's this information is representing? Two, you've always heard about PCR chain, COVID tests. When we talk of the polymerase chain reaction, what are we looking at? Is that a way of amplifying, increasing the amount of the substance from where we need to make samples? Why do they take this long? And three, why are they so expensive? One of the tests that the university used in testing for COVID-19, if you're to test positive or negative, the test is referred to as polymerase chain reaction test, in which we are looking at samples of the DNA that is extracted from a person who's expected to have the disease. But our question is, what does it go through to be able to extract that information out? That's what we call polymerase chain reaction, purposely to amplify, increase, have more samples of the information. And three, when we talk of DNA electrophoresis, what is its purpose? What is the use? Why is it performed? So let's focus on the slide and look at that. When we talk of DNA profiling, do families clash over ownership of the child? What about forensic screening? Crime scenes. Who is judged rightly or wrongly? So, to make it a bit faster, we are moving in this direction. Put it together. We do not need so much about the technicalities and the information about COVID tests and how they are done and what exactly is happening there. But we can have a simple overview that one of the stages is to produce large quantities of target sequences of DNA. Now the question has to be, which types of DNA are we looking at? So if you look at the design in the slide, like we have been seeing before in Senior 5, a double strand, stage 1. Heat, separate the DNA. What does this mean? Are we breaking the hydrogen bonds? Do we need the primers, the starters, to allow the bases to be rearranged? So finally, at the end of the day, our extended nucleotide is added here, and we now form two copies of the DNA. But when is this used? When is it important? How much of the DNA we have will determine the nature of the test we need. If we have large samples, if we had large content of DNA, then we don't need this stage. But they're telling us only when a small amount of DNA is available. It's very hard to get enough of the trace of the DNA. For instance, on a crime scene, samples of blood, DNA in semen, if you're looking at rape cases, tissues that are left in places where maybe a person has been living, hair, how much of your hair has been at the back of your seat in the vehicle. Can we trace all the owners of the vehicle from the time of manufacture? Or if you're found dead in your car, are we able to use the seats as sections to look for evidence to see who else could have been here? And very many others. So, one, we are heating. So we denature so that we are able to separate the strands, like we said. And then annealing. The DNA primers will attach to opposite ends of the target sequence, the places where we specifically require a multiplication. For medical researchers to be able to understand clearly the nature and replication of some of these viruses, samples are got from patients. And then they target a specific area where we have taggers. We tag certain positions where we want to study that gene that possibly is used to manufacture the tentacles, the attackers, the antigens of these germs. So that if we are able to manipulate and study that information, then are we in position to look for components that are able to lock heads with the receptor sites of these viruses or bacteria? So that's why these samples are needed in patients. So once that is done, then we need to elongate. So we carry out polymerization, producing two identical copies of the DNA sequence. Now, when you look at the ratio in terms of what do we have and how much have we got out, we can yield so much from where we are able to make enough of the cases, enough of the studies, to be able to have a variety from where we can get the sources. So, simply put, here, our PCR, copying a segment of DNA, but not a whole genome. We are targeting a specific section of the DNA, so we are actually picking a section of genes. Like we just talked about. What do we need? Why are these results very hard to come by? Why are the exams expensive? Look at what we need. The nature of the lab and stuff like that. So, when we talk of profiling, this is not only, when people think about gene profiling, all they think about is crime, identity, whose father, whose son. It is cutting across the whole spectrum of the world of science. Multiply small samples if you're to study disease. Multiply small samples if you're to look at microgerms. Multiply samples if you're to look at the nature of the genes in these organisms whose DNA section is actually very short. 2, if I was to have an infection, it would be very hard for you to depend only on a small sample extract, maybe from my mucus, from blood, because what you get is a very small representation of what I have inside my body, myself, the infection, and all the new proteins. So to study how these new proteins in the germs are forming, we need to understand the information. And so here we go. So we need, therefore, to carry out polymerization, a chain that continues to produce more and more and more and more until we have so much forming. So you hear about COVID results taking long. Yeah, it does. A lot of procedures, a lot of precision. Now, when we are talking of the profiling, what exactly are we looking at? So we first get our samples, we multiply them. And after multiplication, we need to cut, because we are looking for certain specific genes along the length of the DNA. So we tag from here to here. This is our gene. It dictates for formation of a certain receptor site of a cell, of a germ. So if we are, therefore, to understand exactly how these receptor sites are designed, then it means we need to go back to a very specific section in the DNA, extract it, read it, see the nature of the protein it makes. Now, once that information is available, what else do we need it for? Can medicine be based on that information? Are we able to make substances that are acceptable to these sites of these germs for whose information we have just seen? Now, once we've done this, how do we compare this type of bacterium with another one? If they are all bacteria and they all cause disease, are the diseases the same? 2, coronaviruses have existed for quite a long period of time. The question is, how many species do we have? How much of the similarity of their DNA or RNA do they have? Do they have the same physiology? Are their attack options the same? So what I'm trying to say here is that, look, compare sections of DNA between individuals in order to determine paternity or relationships as evidence in criminal cases, or to identify species. Wow, I love that. Or to identify species. So we talk of disease, we talk of microgerms, then we are here. We talk of who is the father, who is the mother, whose daughter, whose son, then we are here. And then, you're a criminal. We don't need to keep saying, yeah, the next day or in the evening, someone is dead. Is that evident enough? What if I was putting on my brother's clothes and you mistook me for that person? Now, can we go a step further and make the innocent fully innocent without a doubt? And there are some people in prison, in Majirajira and other places, actually there, innocently, serving a sentence of someone who is a criminal and is still with us. I think the judiciary needs to think about that. Let's go that far. So as you can check, the bed has charges, so we have an electric supply. So the plate that is embedded in is going to spread out. As you can check, a fluorescent marker binds the triplet in the DNA fragments, so the results can be seen. Samples added to a gel electrophoresis machine. It's a chamber. Heavier fragments will stay close to the origin position, and smaller fragments will go further. So we look at the spread. Which ones stay bottom? Which ones are spreading up? So the pattern that now we do have here shows up for each DNA sample, and then we start comparing them. Okay, so we have this plate, then we have another one. So if we put a sample here, and another one here, and another one here, and they start spreading through, like the way we do chromatography in senior 1. It operates under the same principle, only that here the difference is the electric charge supply. So, there you go. Take a sample. I know most of you, those who are at home, you know this small stick. Multiply the DNA so that we get enough of the amount. 3, restriction. Identify certain sections where we have specific genes we need. The cut sections embedded in. Once the whole process is over and current is passed through, so you see the samples, sample 1, sample 2, sample 3. At the end of the day, when the current passes through, how is it going to appear here? Number 1, number 2, number 3. Now, let's look at the lines very carefully. Do we have any link? Yes. Do we have any link? Yes. What about this and that? What about this? Does it have any connection? So if we check sample 1, and then sample 2, and then sample 2 and sample 3, check out here. Think about this before we go. This linking with this, and this linking with this. Could this be a mix of content? Could this show a relationship? So we may be looking for a number of shared bands, e.g., paternity, or total match. I love that word, total match. No doubt you are the one who has raped this person. You are the one who has killed this old woman, because the blood stains in the house are showing us 100% clear match with the blood sample we got from you as a suspect. I don't know why they have to wait for weeks and weeks, and they're waiting for the judge to pass the sentence. Science should have a way of creating things work out easily for us. Hey, crime scene, Zubu. Now, and there we go. You ever wondered whether the children you have at home actually belong to you? Are you going to carry out this test without information of the mother? That's an interesting one. So you're a man, you take your kids to the hospital, you do DNA profiling between you and your son, and you keep quiet. Then you come home, and you start telling them, you start acting weird in a way. Go find your mother, as if you're not the father. So now the question is, why do you go for this test? And how shall you go for it? Are you having doubt with your partner? Or has someone come and brought a child in the morning, you open the door like this, baby is crying on the doorway. And guess who finds this child? Your wife. Comes back shouting, can you remember to go for this test? I mean, can you keep the situation calm such that you're able to go and sort it out very well? So when we talk of DNA profiling helping in paternity and forensic investigations, one of the things we are looking at is this. Your DNA alone is not going to give a proper proof that actually this is your son. Remember, you share so much with your brother. So the two as a couple. Therefore, you have to be there. So no more caveat doubting your partners. If you doubt each other, head to the hospital. No, not even the hospital. I think we have a forensic site or forensic laboratory here. The police department, I hear, has one of the best we have in East Africa. So I think, why delay? Have an agreement, go and do it. So we do have a chance now to say, you know what, okay, no more worries, no more problems. Yeah, we shall always find the truth. But the question is, maybe you want to leave a heir behind and you want to pass on your inheritance. So you want, you know what, I must give to my true child. Or you're doubting whether you're actually the biological parent. Now, when it comes to forensic investigations, this is a very great addition to this. But a lot of precautions. Do you have the equipment, the scientific knowledge? How much of the error are we likely to make even when we are using equipment? So skill is key. Training is key. Because the old fingerprinting may not necessarily be enough. So we might need extra. Let's see, where do these fingerprints come from? From the DNA. Can we have it? So it's like a step further. So with that, let's look at some of these activities. Your camera, your phone, take a snapshot. You fight with person, or you have a situation where you clash with people, or maybe you were forced into having sexual intercourse, which we call rape, which is not expected to happen, and maybe in the scaffolds, your hair was left in the lady's fingernails. Can they use that? Or how many people has this lady actually been involved in? Remember, we said the matching. If this is the standard, so our samples are here. Is it A, or B, or C, or D? So we need to look for areas where we have matching. And we roll down until we find where we actually have a component. So let's check out and see. What is the expectation? If we go for DNA match, then we expect 100% to be truly that you are the criminal. Then we need, so not just about, it should be striking sharp similarity, 100% match. The other one, blood clot. Again, we are looking at crimes. Are we able to find it? Taking a snapshot? Are we able to remove the innocence from the guilty? And then we're able to find our criminal. And there we go. Still, we must have 100% match. Again, we need to look for situations in which we have so much of the lines mirroring that the overlapping bands between the victim and the perpetrator suggest a close relationship. And here, used in a wrongly conviction case, judges, or people who are in the lab, you've created errors and you give us wrong results. We've heard of cases where people have said, I was given COVID results that are not mine. What is happening in the labs? What is happening in the test tubes? Are you mixing them wrongly? Are you tagging them wrongly? Have you ever been given results that are not yours? Can this also happen in crime investigation? And if it does, what are the effects? And who is going to pay for the damages? Government? The accused or the accuser? All right, so there we go. If there isn't any strong match, good enough for us to make a judgment, then we have to review the results. We go for more samples. We start the investigation over and over again. Okay, let's look for our parents. And a question, who is the daddy? How many have you been dating? When did you get pregnant? I hope you've taken a snapshot. Now compare it with this. The zygote is a result of 2 nuclei. So we expect 50 from the mother, 50 from the father. That's why they're saying around 50% match between parent and their own child. So we count the lines and check where that is. And finally, we have your homework. Use the same things we've been looking at to be able to do the numbers. So with that, we come to the end of this lesson. Thank you for following.